Hi, it's Dr. Kathy Miller from Indiana University. I don't usually bring basic science results to you, but I saw some results recently from one of my colleagues that were just so provocative. I wanted to join you in thinking with me about them. This is work from my colleague, Dr. Hari Nakshatri. Dr. Nakshatri took samples from healthy women who donated normal breast tissue to the Coma Normal Tissue Bank, extracted single cells and did single cell gene expression pathways, and then categorized them into distant clusters using the same technology, the same analytic pathway that gave us tumor genomic classifications, but using single cell sequencing rather than whole tumor samples. He then looked at those expression pathways and compared them to the genomic subtypes of tumor cells, looking to see if we could understand or begin to propose what normal cells give rise to breast cancer cells. Now, we as a community had largely assumed that each different genomic classification arose from a different cell type or a different level of maturity. So perhaps luminal A breast cancers arose from mature luminal cells, and luminal B breast cancers arose from luminal progenitor cells. And we all assumed that basal-like breast cancers arose from basal epithelial cells. Now, what Dr. Nakshatri found turns those assumptions on its head. He found 4 normal clusters give rise, we think, to almost all of the breast cancers. 68% of the breast cancers are most genetically like mature luminal cells. The fourth cell type that seemed to give rise to the majority of the other breast cancers was a luminal progenitor cell. Importantly, what he found was that different genetic subtypes of breast cancer had different clusters, different normal counterparts, and each normal counterpart could be found in different genomic clusters. So the world is way more complicated than we had assumed, but this sort of technology, the ability to do single cell sequencing in both normal and tumor cells, gives us the ability to really begin to hone in on where do breast cancers start, and if we can find those cells of origin, we can begin to track those earliest changes. And understanding the earliest changes gives us the real potential to better stratify women's risk and to think about much more specific preventative strategies. Now, this is admittedly early days, but incredibly exciting to see this sort of work come to the forefront. Take a look at Hari's work, see what you think. I think it'll make your head spin. This is Dr. Kathy Miller. I'll see you again soon.